Hello, dear students. Welcome in the lecture series on Unit 1 Genes and Chromosome. I am Hemlata Suryawanshi, Assistant Professor, Department of Microbiology, Pratap College, Amalner. So, students, today we are going to discuss on concept of gene, genome, allele, genotype, phenotype, recon, muton, cistron, intron, and exon. So let us study one by one. Genes and chromosome. Let me introduce first. Genes and chromosome are the fundamental units of heredity. As you know that some of the characteristics which we obtain from our parents, which offsprings obtain from their parents, so they come by hereditary characteristics, as we say that. So likewise, the genes and chromosome, they are the fundamental units of heredity. They control the transmission of traits from parents to offsprings and determine the structure and function of living organism. Okay, so these chromosomes, they are nothing but a thread-like structure which are present in nucleus. So in case of the eukaryotic cell, these chromosomes are present in well developed nucleus. But in case of the prokaryotic cell, chromosomes are suspended into the matrix, into the cytoplasm. Genes genes are the segment of DNA which are located on the chromosome. You can see in this image, this is the structure of cell. Inside the cell, there is a nucleus, and in the nucleus, you can observe such a condensed bodies which are known as a chromosomes. And genes are located on the first of all, these chromosomes they are made up of DNA, and on that DNA, genes are located. Okay, so here basic genetic terms. First, there is a gene. So you can also define that gene is the basic unit of heredity. It is the specific sequence of the DNA that codes for a functional product. So which type of functional product, like proteins or RNA, etc. So see certain key points. Genes are located on the chromosome. So you can observe this is the chromosome, and on the chromosome this part which is known as a locus. So here genes are present, we can say, suppose the particular genes are present in this site. They control the specific traits. So traits means certain we can say phenomenon like hair color. When offsprings obtain exact hair color from their parents, so it is because of the hair color genes. So these genes might be located at this site, at this locus. Eye color genes, so at this site of the chromosome, the genes are present which determine the eye color of an individual. Okay, so they are made up of DNA. Each gene occupies specific position called as a locus. As I told you that these are the locus, means specific genes are present at this specific site on the chromosome. And once again, example given, gene for eye color, gene for hemoglobin, etc. Now the concept of genome. Genome is the complete set of genetic material which is present in the organism. So what it includes? Genome includes all genes, all non-coding DNA, mitochondrial DNA. Means if the cell is a eukaryotic cell. Let's see example. Human genome contains approximately 3.2 billion base pairs. Humans have about 20,000 up to 25,000 different genes. Now, the concept of allele. Allele is the alternative form of gene. Means they define the specific characteristics. So, see here, gene for height. Okay, there is a gene which determine the height of the person. So, in that, capital T, small t, these alleles are present. Actually, capital T and small t are the alleles of the same gene. Here, dominant alleles are there and recessive alleles are there. Now, the concept of a genotype. Genotype is the genetic constituents of an organism. It refers to the combination of the alleles present. So there are different types of alleles which are present in the combined form in combination known as a genotype. So let us see the example. If the traits are capital T, so these are homozygous dominant. If it is a capital T and small t, then it is the heterozygous dominant. Heterozygous, we can say, not dominant. And small t, both, then they are homozygous recessive. Here in the image, you can see that genotypes, which are codes for the phenotypes. When the genes are expressed, then they provide certain characteristics to the individual. So we can say that genotype codes for the phenotypes. Whatever the physical characteristics are there, which are totally depends on the genotypical characteristics. Now move towards phenotype. The phenotype is an observable characteristics of an organism. Genotypical characteristics we cannot observe, but we can observe the phenotypic characteristics. These phenotype characteristics are also influenced by genotype and environment. For example, let's see in this eye color of the person, hair color, skin color, blood type, sleep shape, muscle mass, metabolic rate, beak shape birds, different types of beaks are there, ear shape, and diseases. So, tall plant, short plant, blood group, and all these are the examples of the phenotype. Now, there is one formula phenotype is equal to genotype plus environment, isn't it? On the genotype, certain environmental factors affects and phenotypes reflects that changes or expression of the genotype. These particular type of phenotypical characteristics are formed. Now the concept is a recon. Recon is a very smallest unit of recombination. It refers to the smallest DNA segment capable of undergoing recombination, equivalent to single nucleotide pair. So, as we say that recon is the smallest unit of recombination. So, whenever the chromosomes are there, what happens? There is a recombination takes place either in the arms of the different chromosome or that of the same chromosome. So, that is equivalent to the single nucleotide pair. Let us see muton. Muton is the smallest unit of mutation. It is the single nucleotide in the DNA and which is responsible for carried out the mutation. So mutation at one nucleotide, it can also alter the gene. Cistron, it is the functional unit of gene. We know what is gene. And then the functional unit of gene is known as a cistron. It is the DNA segment that codes for one polypeptide, which is equivalent to the structural gene. And note important thing is that this concept introduced by Seymour Benzer. So you have to know about the names of various scientists. This scientist Seymour Benzer proposed this concept of cistron. Okay, in this image, see, these are the different genes and these are the gene products. So functional unit of the gene is known as a cistron. Now the intron concept is there. An intron is the non-coding sequence within a gene. Okay, as we know that informations which are stored in the DNA, then transcription of one of the DNA sequence segment takes place. Both the strands are there, in DNA. So they get a separate out. So the template strand which carries the information which transcribed into mRNA. And on these mRNA, certain types of introns and exons are present. As the introns are non-coding sequences, they get removed during DNA processing. The introns are present in eukaryotes, so they are not translated into proteins. Now the exon is a coding sequence in the gene. Okay, functional or coding sequence in the gene which remains in the matured mRNA. And then these exons, when expressed, they translated into proteins. So in this image, you can observe exons are in a red color, whereas introns are in a blue color. So these introns are removed, these non-coding sequences are removed, and only the coding sequences are there. So after combination of these coding sequences, complete protein coding sequence we obtain. So now in the further lecture, we will discuss about haploid, diploid, lethal genes, partially diploid gene, homologous genes and heterologous genes. So thank you so much. See you in the next lecture.